progress. And then we'll just have a time for discussion or any kind of questions that anyone might have. Let me share my screen. Okay, so the purpose of this program was to provide some basic information about different types of sequencing methods that could be applied to study the biology of cancer. And biology of cancer is defined by certain characteristics that start changing early on, eventually making the tumor or metastasis kind of affect the whole body. And then we talked about how this biology could be studied using molecular data, and what are some of the approaches to extract meaningful information from that data. Meaningful information could be variants in the DNA, so mutations, and that would be in genomic data like whole genome or whole exome sequencing, or it could be in transcriptomic data where we have expression of genes and isoforms, and we can study the differences between different types of tumors or tumors and healthy tissue to understand what kinds of changes are happening. Now, obviously, the specifics of what kind of data is most informative and what is it informative for has to be narrowed down in a specific project. And those projects that we reviewed were several different types of data sets. Some of them were from cell lines and animal models, where we looked at two examples. One example from the transcriptomics 1 and 2, or the project on PDX models, essentially a tumor from a patient implanted into an animal, and then studied where you have a nice separation between the tumor microenvironment and the specific tumor profile, and those could be compared, but also could be studied as a way to understand interaction between the tumor and the microenvironment. In the context of that specific project, we understood how mapping on specific genomes, for example, mapping on the human genome and the mouse genome, can help us separate the two conditions or the two tissues, and then we can compare them. We also talked about the cell lines, and the cell line project was modeling precision medicine in breast cancer, where we talked about specific methods of big data analysis that include clustering and classification. And the main idea there was to provide an example of how we can train a model once we understand what kinds of groups we have, to find those similar patterns in other new data sets. And so a big additional challenge there was validation. So in the context of validation, we talked a little bit about the TCGA, the Tumor Cancer Genome Atlas, where we have a lot of different available data sets. Another problem that we touched on but didn't fully explore during the sessions was the ability to integrate multiple types of omics data. So a little bit of an example there was provided in the precision treatment of breast cancer project, where we have phenotypic data from testing of different drug efficacy and gene expression data. But also in TCGA, we talked about integration of mRNA, so gene expression, and microRNA, and how we can integrate those. So we talked about how those methods of integration really incorporate some aspects of clustering and dimensionality reduction to understand relationship by association. And then we also talked about the different data sets that are available for anyone who's interested to develop a new project, where we provided you, if you submitted a form where you explained what project you were interested in, we then looked at specific data sets that could be used and provided some guidance for those. So that's the overview of the program. I see there are still people that are completing some coursework and running some analyses. We do have quite a number of participants that have, started working on a project or at least expressed a desire to work on a project. So we are working with those in terms of developing those projects further. And one of the ways that we can do that is by looking at, any preliminary results and discussing how those results could be improved or directed towards a more meaningful analysis. So at this point, instead of just talking about some general things, I really want to understand if there's anyone here that already started working on a project and wants to, discuss where the project is. So tell us about what the project state is and where you are and what are maybe some of the challenges that you're facing. And discuss also the timeline from now until the end of the program, so that we can understand, what are some of the likely next steps that we need to do to get you to a final project presentation at the end of this program. So does anyone have anything to share? Asma, I know that you're still going through some of the tutorials and trying to complete those. Okay, Nish, do you want to share something that you're working on? Okay. What about Mikhail? Did you make more progress since after we spoke last? Would you like to share something or? Okay, all right, that's fine. Asma, did you start thinking about a project or not yet? Okay, I also see that Dahlia joined. So Dahlia, if you are joining now, we just started talking about where we are as a group and how different projects that we did are going. Okay, great. So Asma started a project on T-Bio to reproduce. That's great. Yep. Okay, yeah, so there's really a diverse set of people in this program. Some people are just starting to get familiar with bioinformatics. Others are much more advanced in bioinformatics and maybe are looking to apply their skills to a very specific problem. There are a number of different problems that you can choose from. And we really don't have that much time left in the program, so I'm hoping that we can help you develop the project into something presentable before the end, so that we can have a good sense of, achievement or accomplishment during this time. So the program will end on May 16th. We are May 13th, so we just have a few days left. I know that a lot of you have been working on completing some of the online tutorials and learning about different types of data. Hopefully you started thinking about a project and have made enough progress to at least have data sets that are processed and you can continue your analysis independently after the program ends. You do have the option to continue using the server. We have different subscription models that you can explore. But basically, after May 16th, the program itself will be over. And we hope to have a few projects completed before the end so that we can share that project result as a poster or as some kind of a more of a code line project that some of you have been working on. So it would be great to see where you are before that. Now, I am available for one-on-one meetings before that. We only have 3 days left. So if you do need to talk to someone, to just kind of run the analysis results by someone, get some feedback, please let me know. I could also connect you to someone else on our team that is, for example, more technical from a coding standpoint, or more biological from, the biology of cancer. So please let me know if you have, a need in something like that. And if you do have a draft, there is a way to submit your final project. We will send an email about this. It's actually from the back end, from your profile settings, where you can upload your project. But it's also the same place where you will see the certificate for the whole program. So I'll just quickly show you. I don't know how much all of you have been able to explore your profile. So if you go to your profile, you'll see some of the basics. I'll just explain briefly the requirements for completing the program itself. As you know, your name and your profile are listed here. You have your badge, unless you joined another program during this time, but you should have a badge of whatever program you're in, and then the number of points. And you can always click on this points system to see your progress, so kind of recent activity, different pipelines and projects or courses that you have finished. If you take a look there, we are asking that you get at least to 10,000 points to have a certificate of excellence. And if you have below, but you have completed at least 3 courses, you will have a certificate of completion. So you can look at the different sections of your profile right here. And under your profile tab is where you'll be able to see your program certificate. So it will show up right here. And you can also upload your project here as well. Let me just quickly find that. So here you will find the certificates for your courses, the completed courses that you've done. I haven't completed any. So I probably don't know if I can show it on my profile, but I will. Maybe it's right here. So I'll just show you quickly on another profile what that really looks like. Okay, yeah, so here you see profile, certificates, and independent projects. So here you will get a certificate for a program. You'll also receive it by email, obviously. But that's where you will have your certificate for the program. And here you will be able to see your independent project or upload your independent project. We typically participate, yeah, so this is just an example. So you will get a certificate that looks like this, and this certificate will be good for 12 months. So we do ask that you complete at least 3 courses, and if you get over 10,000 points, you will get a certificate of excellence for doing exceptionally well during the program. We do have a few people like that. And so we ask that you just check for any of the courses that you might have completed to 100% but have not clicked finish course button. That will not give you, the points for completing that course. And also, if you do have some progress with your materials, with your project, you can either develop it as a PowerPoint or a presentation where you review the data, articles that you relied on to collect the data and to analyze, uh, describe your methods, whether you used the platform or you used other methods on your own computer or infrastructure. You can describe what exactly you did, what the results were, so what were the findings, and then some kind of a discussion about what next, right? So you want to be able to talk about how you found maybe opens up new questions that you would like to explore. Perhaps you're thinking about completing the project in the future and combining it with other types of data, or maybe validating your findings in more of a biological sense, right? So you found some genes, you would like to confirm that. And that will get it to a level of a poster, essentially maybe something like a conference poster. So if you selected one of the projects that we offer as a starting point, you obviously can still modify the analysis and go further with your biological interpretation of the results, and that will be novel enough to be able to talk about it. And I think that's an important part of being able to talk about the methods and the data sets that we have been covering during this program, to be able to speak about your experience as a bioinformatician or somebody that understands bioinformatics enough to conduct an analysis independently. And a big part of our program is to try and help people to get to that level, even during the time of this program. Now, we do also offer a transition to kind of a independent research project program specifically, which is going to start in the summer. So it's going to be a short-term summer program where, again, you can rely on our assistance to continue developing the project idea or even completing the analysis itself. So if you're interested in that information, I'm sure that you'll be seeing it from us by email as well. And if you have any interest in continuing to use the platform and analyze any additional data sets, you obviously can also let us know, and we'll be happy to discuss that with you. So I don't know if anyone has any additional questions for me at this time, but I did want to make sure that we have a few days before the program ends, we have a time to meet and have any questions addressed in a detailed way. Not a very formal meeting today, but if we can use this time to make sure that if you're stuck, to get you unstuck, if you're almost there, to discuss how to get you there faster. And if you have any other questions, I'm happy to answer those. Membership expiration date is May 27th. So the program will end on May 16th. Your membership expiration means that you will not have access to any of the resources that we have. So if you want to continue using those, but if you want to submit a project, that will give us enough time on May 16th to review your project and use it for your certificate evaluation. But if you don't have a project, you don't have a plan to submit a project, then yeah, you can just continue using the resources until May 27th. Okay, so Dalia, do you want to share with us your data set? So what did you download from TCGA? And you can unmute yourself if you would like, or I can unmute you. I don't know if, okay. Yeah, can you say something? I think we can hear you, Dalia. Do you want to speak?
Yes. Can you hear me?
Yes.
Hello. I just start to do TCGA data analysis. I use Fire browse to download the data, and
Mm-hmm.
then I use I take information about TB53 mutation from the uh, from the publication, and then I prepare a table for those samples which is mutated TP53 got TP53 mutation. Then when I run the analysis, I couldn't get the the results like the graph for
Okay.
principal component analysis.
So let me ask you a few questions. I see here the pipelines that you have been running.
Yeah,
So the data that you downloaded, is that, so is this genomic data? So are these VCF files, or what kind of files did you download?
it's the gene expression. It's got the expression value.
Okay. So here, let's take a look at this. This is what you uploaded. So for some reason here you uploaded two files. You should have uploaded just one. But let
Mm,
me take a look at one of them just to understand what you're trying to do. Looks like a pretty big file there.
mm.
And then what is this? Is that the same thing? Transpose. So you tried it both ways?
Yeah,
Okay.
I tried to put the gene and the sample name in different position.
Yeah. So it looks like maybe, have you tried running just one table at a time?
Yeah, I try all the methods, but it couldn't work.
Okay, well, let's try real quick. So here you use DSeq2. Let's for a second just check if everything is fine here. We can take this file. Here's the file. So did you just take all of them? I saw that you labeled groups, mutated, non-mutated. So how did you annotate this? Did you look on TCGA about these samples?
Yeah, yeah, I look on TCGA for the main publication. I got
Mm-hmm.
the sample ID, then I extract those mutated from the mutated one.
But how do you know that they are mutated or not mutated? Was that specifically
There is
mentioned there?
there is one table in the original publication. It's
Uh,
linked each sample name to the mutation status.
okay. Well, let's see. I see that the names of columns are pretty different.
Mm.
What kind of cancer is this?
That's for colorectal cancer.
Let's see what happens. Okay, because I see that you see your names of samples are very different. What does
Uh,
N stand for? Do you know?
Pardon?
Do you know what the N stands for?
No. But actually, I use the first letters and the first ID number.
Yeah, but you see some of them have,
Have
this,
for
but then they have this, but then some of them also have something like this. Okay. Well, let's take a look. You have a lot of samples here, 145 samples. So, I mean, something should come out. And you've tried running this before, just a PCA to see what happens?
Yeah, I tried the PCA.
Let's see. So I see you did DSeq, mutated versus non-mutated.
Yeah,
And then here's your PCA trial. But you do get results, right?
yeah, but how I can see the graph for
Oh, maybe, what did you upload here? Okay, let's see. Well, let's take a look, because I think here, yeah, you didn't get results. Let me try this. Oh, sorry, wrong place. What is the error? Okay, so somewhere here, there was a problem. Maybe some data in this table is not numeric. So let's see what could be the problem. Group ID. Let's see if I will get the same error here on mine. Looks like the same problem. You know what I typically do, and this is the majority of the problems that we typically see. A lot of times when you have strange names that are very different and have a lot of, dashes and things like that, could be a problem. What could also be a problem is the names right here. So sometimes to adjust for that, what I would try, I will just kind of show you my troubleshooting process. I'll add the same file, but instead of using the R script, I will try and look at the standard PCA, because it's something that's wrong with the data, right? So either you can, start editing things. So for example, what I would start editing first is here and here, eliminate all the dashes, okay? So you can replace this with, let's say, underscore. Okay, because you also say non-mutated with a dash here, right?
Yeah.
Yeah. The other thing is here, maybe the question marks. Okay, so we can take this.
But this is a name of genes. It will not be affected if we change it all to the same.
No, these are two names. So this is actually, oh, it's doing something weird. I mean, this is actually, so if you look, let's say this one, this is one name and this is an alternative name.
Yeah.
So
So
if
we
you
can
look
leave
this,
the first one and then eliminate the other.
yeah, you can, for example, just leave the second one. But you can replace them with, some letter, I think would be better. So let's see, this one actually does some standardization of the table first, so it might fix the problem. So in this gene, you see gene ID 29974.
Yeah.
So this is the Entrées ID, and this is the common name. So in each one, so that means that these, for example, don't have a common name. That's why you have a question mark there.
Yeah.
So let's see. Let's give it some time to process. So then you tried to do DSeq to compare the mapped versus, or the mutated versus the non-mutated?
Yeah.
Which is this, right? Oh, this is PCA. Let's see. So let's just check what's wrong with this one. There. Every gene contains at least one zero.
Mm.
Okay, so this is the error that you got.
Mm.
Numeric variable with integer values. Okay, so let's see, first of all, this should be finished now. There, so now we got a result. Take a look at this PCA. Okay, so here, let me just do this. I will just replace this with this. Because I think that everything apart from here, maybe it's because you have some that only have zeros. Let's try and filter those out too. Maybe that's the problem. And then, well, just to be sure, you know, we don't need this. So let's just delete this for now. And let us make sure that all the zeros will make them. So you know what we can do? We can actually change all this to log scale. And let's see down here.
Can we do the normalization or this data from Firefox already got normalization?
Well, no, I don't know which one you downloaded. Typically, they allow you to download FPKM, right?
Yeah,
Is
that's the one I
that
use.
So I would say that for now, let's try to filter out, or we'll see actually if anything has, you know, any kind of a weird thing here. So like this one is 0, right? So we don't need it. So let's make sure we select all except for zeros. Let's see, 12, 152, and how many do we have here? 20. So you see you have over, 8,000 rows that are all just zeros. So we filtered out the zeros. Let's save this.
So you mean the zeros, it will affect the final results if it's all zeros?
Yeah, I mean, typically you filter out the zeros. And, I don't know if there's some kind of weird numbers here. I mean, I see some samples look a little bit strange, but let's close this for now. Okay, let's see what we got here. So here we have your PCA with all the data. And the PCA looks kind of weird already. You can see that there's probably some samples here that are very different from the rest. So to do that, what I would do before PCA is let us create. So we'll go to utilities. Let's upload this new data set. Oh, you know what? The other problem is that you still have group here, so you shouldn't have group.
Oh, yeah.
So let's go. Well, yeah, let's go to. Okay. Okay. In the meantime, when you do differential gene expression,
Yeah.
We have a lot of samples here. Okay, so we need to
Yeah.
drag and drop a lot. But you see this?
Yeah.
This means one of your columns is empty.
Mm. It's without name. It got some
Yeah,
value without sample name.
right. So let's see what happened there. Sometimes maybe you add, space at the end or something. So this, see?
Yeah.
So what is this? Are we missing something?
Yeah, I think the name of the sample is not.
So let's
No.
delete it. But also, if we look at this one where you have annotation, is it also annotated by group? Oh, so that was Max. Okay, well, let's try this. And another thing that I would do, well, we'll try this. Let's see if the other pipeline finished. See, there's your, oh, something again went wrong.
Yeah.
So what did we upload here? The 2. So I think it's just a matter of the files that, we need to look more closely at the file structure.
Uh-huh.
Um,
So there is a
let's,
specific format for that structure which is acceptable in the platform, or I just have to remove zeros and
yeah, well, it's basically, you saw the errors, right? So the errors that we saw here, right here in DSeq,
yeah.
is that there's some error to estimate size factors for a matrix.
Mm.
Um, so I think that's where a lot of zeros was confusing to it.
Yeah.
You also can't upload files that contain group names for DSeq and stuff like that.
Mm-hmm.
So you got to make sure. And then in the other one, in the PCA, we saw that it was failing because some of the cells did not contain numbers. They contain some other information too.
Mm-hmm.
So here you see X must be numeric.
Yeah.
So whenever it was trying to look at your column means, it had problem with that. So I don't know exactly, maybe there's something else in there that's not numeric.
Mm-hmm.
So, exactly what, so let's see, right now, we shouldn't get that problem. Yeah, so we don't get that problem. It's a lot of files to drag, so it's a little bit unfortunate. But also for differential gene expression, um, this is, probably something that you don't want to do for all the genes. If you have genes that are below a certain level, you can also filter them out. So for example, if they are below, probably 5 or 6 on logarithmic scale, they're not going to be informative. So I don't know how many I have here. Let's see, still a lot. But just to technically make sure, so there are 2 problems here. One problem is, let's make sure that technically files are working. And we just have to mess around with the files a little bit to make sure that they are. The second one is to talk about the analysis itself. So can you tell me a little bit more about what you had in mind for the analysis? So you wanted to identify differentially expressed genes based on P53 mutation?
Yeah.
And so you took 100 and some samples, and the only differences between them is that some are mutated and some are not mutated, or are there a lot of differences?
The only difference I use is only the mutation status
But
of
these are
the.
patients, so they're all the same age, they're the same race, everything
No,
about them is
I
the same?
didn't check other things.
Yeah.
Or maybe I can do some grouping for them.
Yeah, I think it would be better to do some grouping because, I don't think it's likely that all the patients are going to be exactly the same.
Yeah.
And so you are trying to understand what exactly?
Well, I am doing actually a PhD project about B53 pathway, and
Mhm.
then I want to know if the mutation status has some link with other genes. So
Yeah,
I
because the
can.
problem here, you will find some differences, right? And those differences, you don't know why they occur. If I'm just going to randomly basically put them in different groups, and you will find some differentially expressed genes. What kinds of conclusions can you make about them being statistically, different between means of samples? I'm not sure that there's really a biological conclusion. If you want to find what other genes are involved in biological differences between samples, you first have to start with very specific, samples that look almost exactly the same. Uh, I would probably say that this is a basic biology question. So studying a basic biology question with TCGA data is probably not a good thing in the first place. Because cell lines, for example, they're grown in a very controlled environment. So you can say, they were grown for the same time, the same environment, they were given the same food, right? Everything else is the same. With patients, you have zero control over those things. You're basically going to be saying, maybe they ate different things, maybe they slept differently, right? You don't know why the difference is there.
Yeah.
So is there a different, maybe a different data set that you can select for this project on
I
NCBI, for
have
example?
I
Have
have
you looked?
I have other data sets from Array Express website, but it has only the expression level of the genes. It doesn't get the mutation status.
Okay, so have you looked on I mean, here's something. Transcriptional consequences of wild type and missense mutant P53 in isogenic human acute myeloid leukemia. So this looks pretty close to what you're trying to do, no?
Yeah.
Why don't you look into this? This is a gene expression omnibus data set, which means you can download the table of expression.
Mhm.
See, you can click right here. It's a pretty
But I
big
think it's not
file.
only uh, cell lines. It's not the patients, or it's okay for.
Well, it's better for the question that you're asking, because the question that you're asking is whether mutations have impact on gene expression in
Yeah.
specific genes, maybe belonging to some specific pathways. So if you can identify, those pathways in patients, again, the problem, you're not going to know why. Is it because mutation or because something else?
Yeah.
In cell lines, you will be able to look at just, you know, that's the only difference between them. So really, you're not, looking at anything else.
Yeah.
Okay, so you see here,
Mm.
they have them separated into samples. So these look like sample names, S14, S22, whatever.
Mm.
And so they have multiple tables for each sample. So it will be
Yeah.
a little bit of work on preparing the table, but this is something that you can work on.
Yeah.
Or you can probably take a look. Sometimes they provide the assembled data. Let's see. Depending on what you want to, work with and how much time you want to spend on all of this. So here you see that they're going to just provide you with, what different cell lines they used, right? You can see what they did, uh, how they were stored, etc. And then exactly what did they use to process the data. So you have to look around a little bit, to find everything you need for this. But let's see. They also provide you with the SRA project.
Mm. You mean the original application?
Well, these are all the SRA. So you see, these are all the RNA-seq FASTQ files.
Yeah.
So it's a lot of files.
Mm.
You have here 96. So I'm not sure you need 96. Maybe
Mm.
for your project, you need maybe 10, so 5 with mutation, 5 without mutation.
Mm.
Probably read because you see they have a bunch of different types.
Yeah.
Right? So probably read this publication. I'm just going to give you this link so you can take a look.
Yeah.
There's a publication here. So something maybe you can use to select the correct small number of samples,
Yeah.
prepare a table of expression, then do differential gene expression. And now in differential gene expression, there will be a lot of outputs that you can use to understand something's still wrong here, to understand what are the pathways involved. Now it does annotation for you of different pathways.
Yeah.
Okay?
Okay. Thank you very much for your help.
No problem. Yeah, and if you need help, if you're getting stuck, then just let us know. It would be great for us to be able to take a look and, help you get there
Okay.
before the end of the project, okay?
Okay. Thank you very much.
No problem. Okay, so does anyone else have any other questions that we can quickly address before the end? Okay, Nish, do you want to tell me which pipeline? Maybe just give me the URL to your pipeline and I'll take a look there. Okay, so here you have the mapping stats table. So what did you do? You were trying to, yeah, the problem here, just from looking at this, is that you directly went to cuff diff and cuff diff is going to be in here, see? So this is your result from cuff diff. So here's your result. You see it tells you the gene ID, locus, etc. And then you've got significant, and then all the values right here. Okay, so for cuff diff, the way to get to this output is to go, instead of download pipeline output file, this is something that we can fix. You go to jobs files, right? So you can go in here and then find cuff diff. And then it's going to be gene expression.diff. Okay, Nish? Does that answer your question, Nish? Is that the file that you were looking for? To download what? What were you trying to download? This file right here, gene expression diff? I mean, I just tried it, didn't take a long time. Let's see. It looks like it doesn't take a long time for me, so maybe your connection is not very good, or I mean, it's not that big of a file. Okay. Okay, it looks like it works now. So try it again. If you have any other questions, let me know. You have all of these different outputs right here, so take a look there. And you know, with this project, again, let's just take a look at the uploaded data. Okay, so you took some SRAs. So you know what you're doing, you know what you're expecting to get at the end as a result of this pipeline? All right. All right, great. Well, thanks everyone for joining today. Again, looking forward to seeing some of your project. Even if you have a draft and you want to share your draft and take a look at it together, I'm happy to do that. We have you know, until the end of this week to figure that out. If not, and you are happy with what you're doing and getting, you know, that's fine. And we will kind of start putting up, you know, whoever did however many things and progress and points and all of those things into some kind of a report for the end of the program. So we'll start working on that next week. So if you want to incorporate some of your progress before we start doing that, try to do that this week. And if you need any help in getting things done, or if you are interested to continue after the end of the program in some other program that we have, again, reach out to us. We'll be happy to help you with that as well. In the meantime, have a great rest of your week, and looking forward to interacting with you on the platform. Thank you, everyone. Take care.